I'd like to welcome you to this session, and as you might know, or you need to know that this one is recorded as all other sessions as well, and I'd like you to propose questions whenever you like to, or in the end of the presentations. Um, I'd like to first of all present Patrick Murigu Kamauniage. He's a senior research scientist at the DTU, Technical University of Denmark, and he holds a PhD in food microbiology and biotechnology, a master of statistics, and one of food microbiology and biochemistry. He is involved in research, teaching, capacity building, integrating microbiology, microbiogenomics and mathematical modeling to provide innovative solutions and tools. And Patrick will presents a presentation. Patrick's presentations. I'm sorry, Patrick, will give us insights in AI in tracking and assessing food path risks and drivers, their application and needs. Patrick, the floor is yours, and you decided just to stay, right? You need this one?
So should I sit here?
No, you can stay here.
Yeah, all right, stand, sure.
Shall we switch that off? It's on. It's on. Okay.
Yeah, thank you. It's in the afternoon, maybe if I stand, it's better for my blood flow. So, thank you everyone for attending. I'm going to present on data needs. It's a data conference, but based on our case studies on tracking of sources of microorganisms, but also the risk assessment and looking at drivers, what might be driving them. The three case studies that I present are on risk assessment, and on another level, out of the microbe, we have the niche where they live, the sources, animal sources, but also on the global level, what might be driving these organisms or microbial resistance from country to country or from place to place. So the perspectives at the end, yeah, thanks. At the end, I'll give into perspective in terms of I ask myself, where can we place this in the food industry? In terms of food industry is not one generic fit, one size fit it all. They are in the really continuum from the really highly controlled to the really highly informal settings. So the first case is based on this publication on risk assessment using whole genome sequencing data that we did. And the aim is to predict right from if you are given the genotype of an organism, can you predict what will be the end point in terms of infection? There are various challenges statistically in terms of sample sizes. Whole genome sequencing is still picking up. The sample sizes are still not that large compared to the genotype in terms of the number of genes. So we have really start challenges in that we have methods that have been tried before, but they average out if you have interactions between the genetic elements, they average them out, and regression models, of course, face the same problem of dimensionality. So many predictors like MLST, gene by gene, SNPs, against the number of samples, which could be very small. So, so the solution that we found, maybe we can use machine learning. That was in 2016, and it was gaining grounds in the spam filtering and other areas. I pressed the wrong one, the back one, yeah. Yeah, so it has been introduced, the applications, and we found that this can really handle the complex structure and whole genome sequencing data. And we have it can be used for many applications. I'm just pressing, okay. Yeah, so in terms of classification, you can predict sources of infections. in terms of regression applications, in terms of unsupervised, where you cluster the data like phylogenetic trees and see where maybe clusters might be related to, and in terms of policy, where you have reinforcement learning. Um, so the first case study, we learned lessons on data. Not that every time we have much data that we have better insights. And we had 10,000 Listeria monocytogenes data, none of which was really important for our models because we need reproducible endpoint associated with it. We had lab studies that study proxies for virulence, but we found quite a nice data set on only 207 strains with 39 clinical ones from food and food environment, which had associated clinical frequency of illness that we can use as an outcome in the model. And then this has, and also food data that we can also explore the models trained on clinical isolates later. But you can ask why these 207 is because of the representativeness of the sampling, because in France, the Listeria is notifiable, so that a national reference point very uniformly collects all isolates both in food and clinic. So this was an opportunity data set that we found to train supervised models. Supervised models are those models you train to recognize patterns, illness frequency based on the maybe the genomes or proteins or something, and then use this on a testing set, and then have an algorithm which, if you have an isolate, you can draw into the machine quickly, and it can predict how much, what's the likelihood of that, or the frequency of the illness that could be caused by this isolate in the population. So this is a flow process. We explore data, cleaning, processing, looking at the genes which are more important. I'll come back to this later. We always have a lot of data that is not offering information enough. And then testing different algorithms. I'll come back to that in terms of data considerations. And then random forest model was selected, which was quite a bit accurate. And from this model, is where I'm saying less is more, is in that the top predictive genes that predict really high probability of illness at the high end. We had a few of them, but they have some biological meaning. From literature, you can tell that yeah, these have been reported elsewhere to be really important. And then using this, you can zoom in and link this to source, maybe the most important sources, by looking where are these genes occurring a lot in high frequencies. And we found vegetables, ready-to-eat foods, of course, clinical, which was just a control in there. And these are foods that are quite associated. So in this case, we can link that and ask ourselves, can we use the same principles to look at the niche where these organisms coming from that are affecting the humans, looking at different foods, travel, environment, and other sources. And then this can help to really have priority on where to intervene. Again, based on whole genome sequencing data, you have different data opportunities, and there we have to compare. And there are many opportunities, like even you could think about, is it a non-protein coding, protein coding, and you know, but then you have to just to compare the different sources, outputs of the whole genome sequencing data, and evaluate them on machine learning for prediction of sources of human infections. So the workflow looks like this. It's just I broke it down so it can provide some data lessons. In that feature reduction, among the, maybe if you look at the pangenome, like 3,000, 4,000 genes, then you find that if you look at the variation provided by each of these, you might find that it's only 200 also that are quite important. In our case, the whole genome MLST only about 200. So sometimes less is not all big data is big, but also class imbalance when we sample. A lot of the sources, eventually the totals, they are not the same. We should not force them to be the same. It can be natural variability in that maybe Salmonella campylobacter is not associated a lot with the cows. It might be with the chicken or pigs or something else, but it might also be with the programs that and in terms of we have agreed that we are focusing more on maybe pigs or something. So this imbalance is bound to be there. There are analytical methods for that, different methods, but also to test different algorithms, not one size fit it all, in that the structure of this data will differ and the methods will be accurate, more accurate depending on the structure of the data. So eventually we train the models on the human Campylobacter, Salmonella in this case, to predict the human cases where these the animal cases, sorry, where these humans were infected. So you train the model based on the food and animal data, and then feed the human data and ask, based on your knowledge of these animals' genomes, as Salmonella from these animal genomes, where are these humans most likely be getting infected from? And this is just a probability scale from 0 to 1. And you can see the big color here is Danish pig. And we have important pig. So pork was really an important source of infections. We evaluated also cattle and others. In this case, pork was important as a source. And I also mentioned that the prevalence also and consumption also might also influence this, because it's a main food also in Denmark as well. And then we evaluated models with the lower number of loci which are selected, the most important ones, and the reduced also high, we can call it highly discriminatory with more loci. Out of the thousands, when we reduce them to 125, but we can also reduce them to 20 most important, and we have a more accurate model, but which in principle agrees in terms of the picture, in terms of the pigs might be the main source, but here which also shows that not all big data is big and important. Some of the lessons that we learned about data from this source attribution were that it needed a lot of people from different disciplines coming together, and bioinformaticians to analyze the data, microbiologists and everyone. Then it also needs to be really also controlled in terms of the year and the places that you sample. You cannot generalize beyond what you sample. So that's a lesson. And then sample imbalances are sometimes natural or sometimes due to other issues, but also we need a common data standardization in terms of the metadata when you are sharing. And also the platforms you sequence from, and this was also published. Finally, we can ask ourselves overall what might be the drivers that we might also regard. And this case study is on antimicrobial resistance based on metagenomic analysis of urban sewage from the first study here is 78 countries. This one is about 105 countries, to look at can we identify some patterns in terms of things we can change to address resistance. The biologically antimicrobial use has been traditionally, of course, even now the Darwinian provoker or agent that leads to resistance. Antimicrobial use, you use a lot, you have a lot of resistance. But there might be other drivers that once you spark a fire, there is a source of the fire, but also there is what causes the fire to spread more. So there might be other drivers that lead it to go to spread and more. So we look at the indicators from World Bank datasets, and also in terms of antimicrobial use, we had lessons on data here because it's really not all agents that collect data collect in all the same countries, but there are also bits of overlaps that can enable you to infer how much agreement are these data sets. If they collect in maybe 10 countries the same, you can look at the regression line and see how they are agreeing and get a new antimicrobial resistance measure that can be now generalized in all of them and be used for modeling. And then again, the issue in data is also missing, a lot of missing data, a lot of highly correlated data. Again, not all big data is important. You have high correlations, and these ones you can use unsupervised methods, hierarchical clustering, to get manageable bits from that. And again, we had quite a nice model here that can be used to predict those countries we don't have sampling, but also to look at the important predictors. So in this case, the model was there was quite agreement between the predicted and the observed. And it's all in agreement also with the epidemiology of resistance, in that this belt here, a bit in the south, we have quite high resistance. But if you look at the most important variables in this, they are related predictors of higher resistance. They are related to those variables that indicate poor healthcare access, wash, like defecation and diarrhea, like wash means water, sanitation, and hygiene. And those that predict lower resistance, they might be related to better access in all these. So there was some, and the World Bank also cited this. In some new ways we can look. This is not causal. We did not plan the experiment to be causal, but it's indicative of a hypothesis that we can pursue in terms of where we can do. But when we look at the data, metadata that comes with the sample, in terms of when you are sampling, it's very important to collect even some information, like even the seasons, the use. We found that like here, the seasonality was quite important. Although use did not really turn out as important, but seasonality is important. It might be to do with the spike of infections, so it might inform you something about the good. It's really important to collect data around the samples themselves, pH, maybe other parameters of the samples are quite important. And then, but we also have to recognize that, of course, for example, not all countries use the same antimicrobials, because not all countries have infections, priority infections the same. And you have characteristics in where the samples are coming from. This was sewage. They were quite different, but also infrastructure, but also the countries and the regions differ a lot. So this heterogeneity should be considered when thinking about the samples. For example, this is just one case of phenol resistance. If we look at the potential predictors from this World Bank dataset, we see that if we divide the countries based on finite Misha modeling, which is a different hypothesis testing into clusters, how countries cluster, then we can find that whatever is positively, if people are living in slums, you have higher resistance, but not in all countries. This is a zero line here. It's not showing somehow. So it's not all countries that living in slums will be important to. Of course, we know that it's associated with a lot of infections because of water and other things, but not in all countries. So we have to think about the situations, the scenarios in the country, and in different countries or region specific, and even the policy maybe should be specific for the context. And then, but something else we should know is that these data sets, they look good because in the website is really good. But if you look in the details, that's where the devil lies. This is a website, one of the variables, one of the 1500. This is physicians, which was a quite a good predictor, is well collected. Number of physicians, a lot of organizations collect this data. But if you press on the details, they also give a disclaimer deep down in the document that data comparability is limited by differences in definitions and training on medical personnel will vary. Not all physicians are trained the same. Not even human resources are also focus on the urban area. So it's really important to look also in the details that are deep down coming with the data also. This might also influence a lot. But in terms of the, from my previous experience, in terms of industry, food industry, if we now bring the context issue, not all companies have the same food safety management system because there are many of them, and the stakeholders who are imposing this are many and different, and they impose them differently. And the environment is different in terms of what are the priority pathogens so really, even when you look at the data that might be important for a small scale producer is really not important for a multinational. So we might have to think about that. That the product safety culture in the industry or even in the farm, wherever product itself, and even organizational, how is it organized, and even the level of food safety management system. For instance, if you look at it might be on a continuum from law, something that is totally is a guy who is starting, they focus on the product and its selling, but so they don't have a good food safety management system. It might lack, and someone who has it has copy pasted, so it's based on best practice. Then you find a guy who is even having an R&D internally, and it's really scientifically underpinned. So for all this, if you need to predict food safety problems, the data scenarios will be different. But how to do this, you need to look at the contextual, right, from the context. What are the product? Is it milk, and is it meat or something? And what are the challenges with that in perishability and pathogen? What about the processes? Is it a kill process or is it a minimally process? What about the organization? But also the environment where they are working, both the physical but also regulatory environment. They put pressure on the food safety management system, and this will affect the product quality, even the microbial food safety. So when collecting this data is quite important that we also think about the data around the data, and yeah. So that's all for me.
Thank you, Patrick. Is the microphone on? No. Thank you very much. Do you have questions? We have 5 minutes for questions to Patrick. For now, then just one comment. Thank God we're working on metadata and data management on the data collections in the EU. Giovanni Yokono, I've not introduced you, my rapporteur. Do you have a question?
Hello, I have a question that relates to the first part of your presentation, and I was thinking that now the risk assessment is done for a microorganism looking at the presence of genes of concern, no? You also mentioned databases, AMR, metabolites, and so now you go through the genome and you search how many genes of concerns are there, and then experts evaluate the implications. So can you tell a bit more about the comparison to do things as I just said, or just feeding an entire genome or a list of features and getting an output according as you showed in the initial part of your presentation? Yeah.
mean, involvement of experts to validate the
No,
or,
not so much the experts, but not because of the experts, but comparing the idea of just going through the presence of genes of concern,
yeah.
or just feeding the whole genome to this ML, machine learning, and getting a reply of a probability of some
Yeah, I would.
illness, and so.
Yeah, I would also now is improving a lot. Even the presentation in the morning, if you caught it, there are methods that also use weak predictors, many weak predictors, also like bagging algorithms that boosting algorithms that also use many weak predictors and enforce the prediction. So if the purpose is prediction, the more the merrier. But also, and this comes with an expense, the cost. If you can only afford maybe multiplex PCR, maybe these genes of concern can be also very important quick indicator. If it's fast, if it's less costly, then the reduced dimensional genes of concern, that would be really important. But in terms of predictive power, I think with the cost of whole genome sequencing going lower, I think the more data that you have the better.
Okay, thank you.
Thank you. Thank you, Patrick, again. Then I'd like to introduce to you our next speaker, which is Pimplapas. Lehit, Sharon, and Fon. Very good. Pimplapas has a position as senior researcher at as well DTU, and as you might have recognized, they're both colleagues at the Technical University of Denmark, and her position is equivalent to an associate professor. She holds a PhD from DTU. The topic was whole genome epidemiological typing of Salmonella, and has a master in bioinformatics. Pimplapas' presentation will be about applying sequencing and machine learning for improving food safety, and she will present a recently developed machine learning tool to analyze VGS data. Pimplapas, the floor is yours.
Thank you for introductions and thank you for having me. I'm also talking about another kind of data that I utilize and use it for machine learning or AI, but data that I'm talking about is only DNA data. So it's just probably different from what you have heard from the rest of the day. So this is a project that I'm trying to apply sequencing and machine learning for food safety. So you can see the question that I'm trying to deal with, the question in the food safety. And what I'm doing is, but first of all, before going to the detail of my project, I want to talk a little bit about the AI, or something that you already know and use a lot, the ChatGPT things. So this is like the AI, they are using our language, everything in the internet, our language, text, whatever, even our speaking languages, and trying to feeding in learning from it and spread out the answer that we want. So they basically learn from our language and give us some specific answer that we ask them to. But actually, there is another data, another language. It's called DNA or RNA, which is actually a universal language. Every living organism speaks these languages, either DNA or RNA. So if we look at DNA as a language, why not this technique of AI and machine learning can learn from this data of language, universal language, finding pattern, learning from it, and giving us an answer to some of the specific questions. And that's exactly the idea of why we applying this machine learning with this bacterial DNA data, learning from the pattern and giving us some of the answer that we want. And what the answer that we want, we aim for this organism, which is very much problematic, one of the problematic bacteria in food industry, especially in the dairy industry, which is Listeria monocytogenes. They are something like they're always there. They're always there, never gone. And some of them, they are more virulent than the others. Some of them also can tolerance to the disinfectant that the company actually use. So my questions that I want this machine learning tool to answer to me is, if they found a Listeria monocytogenes, how dangerous is the found pathogen, and how they get rid of them properly. So So that means I want to know the virulent potential, and then if it's resistant or tolerant to disinfectant that they use in the food industry. So this is the basic two questions that I tag on, and that is one of the most important food questions for their food safety. So, by the way, Patrick also give you some of the machine learning model on this virulence prediction. And actually, we also like a continuation of the model that my PhD, Alexander, and my postdoc, Mirana, they are all former postdoc and PhD now, they're working on this. So let's go to the first model on the predicting virulence. So in order to use sequencing for machine learning, of course, you need to have sequencing data. And we have whole genome sequencing data of the Listeria monocytogenes. And then, not enough to have sequencing data, you need to have an outcome of the sequencing data. What is it? Of course, the Listeria, but what is the virulence potential of that sample? Okay, and Patrick also giving you a little bit of the virulence potential that we use. There are many other ways that you can, identify virulence. You can use the animal models, that kind of thing. But I can see that in the food industry, they will not going to do an animal model in their lab. So we come up with another predictor for the virulence. We call them virulence potential. And that's not actually we, it's some of the other previous publication. What they did, they took Listeria in the same year, in the same reign of time, number of clinical isolates divided by number of clinical isolates plus the food isolates. So the more clinical isolates compared to the food, that means they're present in more human. That means it's more clinical frequency, and that for us, it's more virulent potential. And that's why we came up with this number. And instead of predicting exactly number, we divide them into less than 50% lower, 50% 70% medium, and more than 70% higher. And because it's easier for interpretation, if you give the model, give the user is 70% clinical frequency, they wouldn't know what it is. But if you categorize them into like a traffic light, lower, medium, and high, that is making sense in terms of interpretation. And if you look at the distribution of the strength that we use in the training, it's kind of, we have two sets of data. I'm going to talk about this later. But look at just the national surveillance data that we got from France and Denmark. It kind of makes sense that the highest virulent potential isolate, they should have less number than the other ones. Otherwise, we're gonna, infect it a lot from Listeria monocytogenes, right? So this is kind of makes sense that we divide into something like this. So talking about the data set, of course, the higher the better. But when you talk about the higher, sometimes it's coming with some error as well. So we started with a national surveillance data. This one is the data that Patrick already showed previously from the previous publication of the French national surveillance of Listeria monocytogenes between 2014 and 18. We also had an access to our, Listeria in our food authority of Denmark. We went to look at the similar years of Listeria genomes, and we got around 109. Those the one that we can use in the model, and we can get the virulent potential percentage. But the thing is, you have around less than 200, 169 genomes for the training. I accept that it's quite low for machine learning, but it's very trustable data. Then we look at the public data. We got to the genome tracker from the US, but where they have all the pathogens more than just Listeria monocytogenes. We took at them in between the same time, look at is it food related isolated or clinical related isolated, and thus we can get the same virulent potential percentage. But the thing is, if you go back here, look at this. So they have very high number of very high virulent potential genomes. That's no surprise because most of the reported, they are human strains. And that's why. But anyway, because the good thing is they have so many numbers of genomes compared to the national surveillance. Of course, we took them all when we trying to build the machine learning. Okay, let's have a look in the infrastructure of building the model. When you look at the genomic data, there are many places, like millions of base pair. Which one, which part of the DNA or genomic that you're going to be using? We also don't know. But what about the SNPs, the single nucleotide polymorphism? Is that going to be a marker for the machine learning? Is that going to be the non-virulent genes? We know about less than 200 genes that confer to be virulent in this monocytogenes. What about all the genes that we call pangenome, pan mean all? What about all the genes in this monocytogenes? This is SNP, this is a few hundred genes, this is thousands of genes. We took them all and test which one is give us the best predictor in the machine learning. Okay, basically, first of all, you're going to transform the genomic data million of base pair into a metric. Metric of what? Metric of either SNPs present absent, or metric of virulent genes, percentage of virulent gene, or present absent of gene, present absent of all the pangenome, thousand of genes. The outcome is low, medium, and high virulent potential, and each row is each of the genome. Okay, and then you divide them into training set and testing set. The training set you feed into different machine learning algorithm. We try every algorithm that is available at the time, and then you test the performance model using the testing data that you divided from your input data. Once we have the best model, that model, we're going to use it as a predictive model. Once you have a new genome of this monocytogenes, you transfer the genomic data into the same metric that you use to build the model, and the model will give you low, medium, or high virulence potential. Look at the performance of the machine learning. Here, if using the national surveillance data, about 200 genomes, this is almost 3,000 genomes from the genome tracker. Luckily, both of them give us, and here it's different machine learning model. Luckily, both of set of data giving us high percent accuracy. But what is good for this, because we compare this to different gene markers in genomic data, virulent genes, pangenome genes, and SNPs. And you can see that the pangenome gene perform the best comparing to the other either SNP or virulent genes. This is only for virulent potential. I don't say that is pangenome going to be the best for everything. Okay, so but at least we know that okay, pangenome is the best in terms of predicting virulent potential. And at the end of the model, we actually end up using the pangenome for predicting this virulent potential. But anyway, this testing is based on our data. It's still our data to test the performance. Then we want to try, what about the other we call independent testing data that not related to our data at all? So we're seeking out any previous publication that they did animal model on the Listening monocytogenes, and they provide whole genome sequencing data and giving us hyper or hypovirulence, and we didn't have many, but even though it's not many, we took those genome that we know the outcome of virulence into our final model, and this is the result. Even though this is the independent data set for testing, the accuracy is still quite high. It's still going to 76%, even though this is the data from animal models that nothing to do with our training at all. So it's like completely independent. That was the model for virulence potential. The other model that we built in our project, this is for disinfectant tolerance, to help them to know how to get rid of this properly in the food production. Okay, the thing is, of course, we need two things. Again, sequencing data and outcome. This outcome mean the disinfectant tolerance or resistant. The problem is there are not many of these data available. If they're available, they use different, lab setting to quantify or identify the tolerance. If we're gonna use those data, it's gonna be a mess up for us because the way they build it is different. So we want a data that we test in the same way, meaning that we need to build our own data for here, our own data of knowing the outcome, in this case, the disinfectant. So, and then I'm going to tell you how do we do that. And then we put this in the machine learning model. My initial idea, I just want to know is it sensitive or tolerant to the disinfectant. But my PhD, Alexander, want to go above. He want to predict the actual MIC value. So not just yes or no, how much the tolerance. That's what his aim. Okay, then let him do that and let's see what he got. First of all, as I said, we need to build our own training data here. We collected 1,600 isolates of Listeria monocytogenes from around the world. That's not really around, but made from different countries, spanning from different times and also from different sources. These 1,600 isolates, my PhD, Mirena, has to put them all into the lab to test in broad microdilution to identify the MIC value and get the cutoff to know if it resists sensitive or tolerant to these three disinfectant. 1,600, that take a lot of her time to doing this, including whole genome sequencing, all of them. And what she tests is with the most used disinfectant called benzene chloride, BC, and also DCAC in the media. This is a commercial one. This is pure substance. This is commercial that they actually use in the food industry. So she tests them all, and she got the MIC value, and she got the cutoff, and she know which one is sensitive or tolerant to the disinfectant. And now we build the models. It doesn't go further. Okay, good. So we did a similar thing that in the machine learning model, as you have seen in the virulence potential, but we found something that it might be worth to take into considerations, especially we have larger data set in this modeling, is these 1,600. When you try to predict them, what you don't want to predict something correctly because they are clonal. They're twin. You predict them correctly just because they're twin, not because of the property that you want to predict. And that's what we take into consideration of the phylogenetic signal that could interfere in the machine learning development. Because if you put the strands that is very closely related, together into one is training, one is the testing. You build a model on the training and you test the performance using the testing. But the testing and the training, they are twin. Of course, you're going to get the high percentage. So not knowing that it is from the phylogenetic signal. So what we did here, we're trying to look at there are many ways that you can look if the strands are similar or not. The easiest way, you can use what we call ANI, average nucleotide identities, that is very quick and dirty to get to know how much they're similar in the whole genome. But my PhD, he used the camera method that he trying to figure it out, how much they're similar, and then they make a cutoff and make sure that those are the similar one won't be in the same, won't be in training and testing. They need to be together, either testing or training. They go together, okay? Anyway, so have a look at what he has. So this is the performance of the different models in the predicting sensitive or tolerance to the BC pure substance. And here is the performance on predicting the MIC values. Let's first have a look at this. The higher the better in here, because the higher the more the accurate, the balance accuracy, the more the higher. You can see that multiple models giving a very high predictive accuracy for either sensitive or tolerance. But when it's to the predicting MIC, the actual values of being tolerant or sensitive is not the balanced accuracy, it's mean square error. So the least the better. So the least mean square error is mean the predict value is not go further apart from the actual MIC value is supposed to be. So the least the better here. And yeah, there are some model that also good to predicting the MIC values from the genomic of the listed genomes. Okay, that seems to be good. Either sensitive prediction of the disinfectant or MIC values. I really have problem with this. Is there anywhere else I can press? Okay, that one is better. Yes, much better. Good. Yes, that's for the BC. We also have the other disinfectant, the one called DDAC, is a pure substance, and then meter. We built the model for DDAC. We did not build the model for meter. Why? Because meter has many ingredients, but the most main ingredient is DDAC. So we assume that okay, we have the model for pure substance. Why not we use it for the predicting the commercial substance that the main ingredient is this pure substance. We try, and this is the performance. It's still quite high. So that means the model also very flexible that you use it with the pure substance, but you also can apply with the commercial substance that contain the main ingredient at this main as a main ingredients. Yeah. Lastly, our model, either virulence potential models or the disinfectant tolerance models, we combine them into a tool that we call ListPred, Listeria Prediction. And the thing is, we include the virulence potentials, we include the disinfectant resistance, and then the user will input their genomic data. And then what the model gonna give to them is a virulent potential, low, medium, and high, or sensitive or tolerance to the disinfectant. We also test the entire pipeline of ListPred with different input data because now the technology of sequencing is not short read only, it's also long reads, right? Nanopore. So we tested the performance of our pipeline using both the same isolate sequencing both in short and long reads, short by Illumina, long reads by Oxford Nanopore, and see how it go. And also, if we input the data in terms of raw read format in FastQ data or assembly in FASTA format, how it's gonna give you. So the conclusion is, it's a very great overlap between either you input short read or long read data. So it's fine, either you short read or long read. It's a little bit of mismatch between raw read and assembly, but I think it's still not that high. So that means that the pipeline is also flexible for both technology, either short or long reads. And actually, in the food industry, when they start to invest in the sequencing, they have never invested before. Going through the long reads investment of nanopore is cheaper than they're going to invest of the short reads big machine of Illumina or the other thing else. So, and what about the program of ListPred that we made? We have made the web version. So this is just freely available. You go to this website, you input your genome, either short reads Illumina or long reads Nanopore. Just one by one, you're not allowed to put like 10 genome at a time. And then the tool will give you virulent potential and disinfectant resistant, but you are a developer, you can get the tools in the Docker format or in GitHub format. This is for a standalone solution. You can install in your computer, in your HPC, and then you can modify the tools as you wish, because this is the coding of the tool. You can use it to modify it or getting better than it is. Okay, so, and I think that's it. This is all the people that involved in this project, especially my PhD Alexander, my former PhD Alexander, and my former postdoc Mirena, that did a lot of great work in this. Also Patrick also here in this project. And that's it for my talk. Thank you.
Thank you very much. Thank you very much for your presentation. Are there questions in the room? There's one. Sabrina.
Thank you. It's sounds really like a great model, and I'm curious if you have it implemented to your laboratory database, and do you get a prediction for every sequence you analyze in the lab out of the box, or do you also put in the sequence to the online tool and ask for the prediction whenever you need it, or is this kind of a routinely thing?
Oh, you asked about our lab?
Yes.
This is mainly that we built it for the food industry labs. So one food industry in Denmark right now, one of the biggest dairy industry, he also part of the project. They use this tool in their pipeline. Once if they have Listeria genomes, they need to know more than just Listeria. So they use this tool to know virulence potential and if disinfectant resistant or anything or not. So this project is aimed for food industry to use it more and more, so they will have better control of what Listeria they have. And of course, if the national food in the national food FDA of Denmark, they also interested in using it. We can easily integrate it. They already have a pipeline for genomic data. This can just plug it in, like a Lego. Just plug it in easily, and they have two more things that they can know. Yeah.
Okay, thank you, because it really sounds great also for the official controls.
Yeah.
Thank you.
You have a question as well?
So maybe it's on? Yeah.
Yes.
That's a really basic question, but you put the whole genome in, you train the system, and then you probably get like a probability of how probable it is that this strain causes disease, maybe. But does it also give you like, it's because of those genes that are present and also because of the pattern between different genes, maybe?
Yeah. So that's called explainable variables. So this is a good question, and I thought that you might ask this. That's why I have it in the backup slides. So this is not the virulence one. The virulence one, Patrick already showed you. So in our previous model, Patrick also showed some gene and that related to virulence. I want to show this. This is the important genes for the disinfectant resistant prediction. Okay, and the machine learning can rank you. So you can ask the machine learning, okay, spread out genes or markers that you use for this prediction, and they can rank them from the most important to the least important. The most important, it doesn't mean that this gene alone will do a great job. It just say that it's more important than the others, but in terms of prediction, you might need them all. Okay, it doesn't mean that you can take the other thing and then leave all this and it's gonna give you good things. Okay, but anyway, we take some of the most important one and see what they are. See, some of them we know and it's good because quark S, they are a few genes in Listeria monocytogenes that we know that they're related to disinfectant in BC. Only a few genes. It's not like in antibiotic resistance, we know thousands of genes. In Listeria disinfectant, we know a few. When I see a few genes, 3 or 4 genes. And then the other ones, even though it's not disinfectant resistant, TET-R regulator. Another one, TET-R family transcription regulator. So this is also another gene that related to disinfectant. Even though it's not disinfectant resistant, it is something a regulator that is related to antibiotic resistance. So there might be another much more mechanism that we just don't know. But my point is, it doesn't spread out just a random gene for you. Some of them also make sense, and some of them we don't know if it makes sense or not. Yeah.
Thank you.
Yeah.
One
Okay.
further question, right? You have one? I have one. Okay, let's see. Are there some from the auditorium? If not, we have two further questions. Do we? Yes.
No, I had a question that was almost completely covered by this last question, but yes, it's the same as you are showing now, but for the virulence. So my question was, for the virulence prediction, did you find, so with the pangenome it was working better?
Yeah.
So the question is, was it working better also because there were some virulence gene that were, let's say, absent, that were not included in the virulence subset, and you found some genes that either they were known but not put in the virulence subset, or new candidates for being new virulence gene? So did you also find new candidates for unknown virulence genes?
I don't have that list of virulent genes, but I can tell you, in the pan genome, thousands of genes, the most important genes around 700. Those 700 doesn't mean that 200 non-virulent genes in there. So it's not like, okay, it must include all that 200 in that 700. No, only some of them. I don't know how many numbers. That means in the predictors that is most important to the machine learning, it include already known virulent genes, but not all. It also contain the other genes that we don't know.
So we don't know if they are virulence genes,
We don't
or
know,
they
and
are
some of
just
them just
normal genes that
hypothetical.
must, yeah.
Yeah.
Okay. Thank you.
That was exactly the question I had as well. Thank you, Giovanni. Okay, then thank you, Pimplapas. I think
Thank you.
there were all the questions, and maybe later for coffee, and then I'd like to introduce the next speaker.
Okay.
Okay. Thank you very much. Okay, then Michael Schneider will is also here for us, and he is the head of the AI consultancy in the KIDA project. Might have already heard in this morning about the KIDA project, which consults employees from several research institutions of the Federal Ministry of Nutrition and Agriculture in Germany. He holds a PhD in machine learning from the University of Munich, and he's well experienced in academia as researcher, scientist, and data strategist. His presentation is about how to prevent food fraud by pimping analytical methods with ML approaches. The floor is yours, Micha.
Yeah, thank you very much for the kind introduction. So I would like to use the time to speak about applied machine learning in food authenticity using the example of cactus oil. So in a nutshell, what's the project about? So we're considering fake oil. So fake oil is an oil which is adulterated with some other kind of cheap oil. And this can be seen as an increasing issue in food authenticity in Europe, because there are a lot of reasons why to do this. So possible reasons are, for example, the bad harvest, also influenced by climate change maybe, then also armed conflicts, and of course there's also the possibility that the companies want to do profit maximization. So let's have a look at the pictures. So how does it look like? We have here a bottle of cactus oil. It's quite expensive, so around 600 euros the liter. And what is done is that some companies might add some kind of sunflower oil. That's very cheap, so just 3 euros per liter. And as you see, this kind of sunflower oil is refined, so it's colorless. So you can imagine if we add this to the pure cactus oil, it will give this adulterated cactus oil, which from my perspective is completely looks the same as the first one. So if you would buy this in a shop, for example, you would have no chance to detect whether it's an adulterated one or a pure oil one. So the question I want to address in this speech is how to detect the oil adulteration with machine learning techniques. And so we started with the data we have, and well, the oil is quite expensive, so we have only a few data points. So that's our first challenge. We have a few data points because of the expensive of the oil, and also the time we need to analyze the oils in their different attributes. So in the end, we have 37 features or attributes, and in different groups. So one is the fatty acids and some others. And the sample size is in the setting 27 cactus oils and 10 sunflower oils with which we want to adulterate the cactus oils. Um, and so, because that's the case we have to deal with, we thought about some simulation techniques to improve the databases. And we thought about two we want to compare here. So first is the Monte Carlo simulation with multivariate normal distribution. So the idea is that we estimate the mean values of the cactus oil, so the features, and also the covariance between the features, and then we draw out of this distribution. At the right, you see an example of of a normal distribution. So that's a bivariate normal distribution. So you get some impression how it could look like for two features. And the other technique we tried out is the conditional tabular generative adversarial network model, so shortly CTGAN, I will use in the following. So that's a deep learning technique. And here the neural networks tries to learn the underlying distribution of the observations and generates data out of it. And so we compared these two models. So here you see the results compared with the Kullback-Leibler divergence. And the first column, you see the number of simulations we generated out of the two methods. Then with MC, that's the Monte Carlo results. And then with CT-GAN, that's the results of the CT-GAN method. And the epochs is the how many times you run through the training process. So the more training process cycles you have, the better the values become. So in the best case, we would like to have a Kullback-Leibler divergence of 0. So then it would mean that the distributions are the same. And the farther the numbers are away from 0, the worse the mapping. And so we see after at 10,000 samples, we had a decrease in our values, which we like because it's closer to the observed one. And the Monte Carlo simulation shows here much better results than compared to the CT-GAN one. To sum this up, it's clear that the Monte Carlo simulation produce results which are closer to the observed distribution, which we observe with our values. And so we use this method for the further steps of how generate the data. So the next challenge we have to deal with is how we can detect low levels of adulteration. So in this project, we have 7 classes in total. So first we have the pure cactus oil, then we have adulteration starting with 1% of adulteration until 9% of adulteration, and then pure sunflower oil. For this, as I said, we use the adulteration, we use the simulated data for the adulteration. And then again, we use 2 different machine learning methods for classification. So one is the neural network and the other one, the random forest one. Just a brief introduction. So what is random forest? Maybe not all of you have seen it before. So it's a supervised machine learning technique, and it built up from decision trees. So here we see an example of one decision tree, which is using binary decisions on feature or attribute level. So we start with a data set, let's say here 1000 samples, the data, and then we split the data in 2 classes, so according to a certain feature. So if the feature of this observations are below a certain level, all observations go to the left side, and if the number if it's above this level, then it's to the other side. And that we can do very often. So here's just an example with a depth of 2. So at the end, we get the classes we want to predict. So here on the left side, we see the pure cactus oil, for example, and then we have also the other mixture classes. So with 1% of adulteration and 3% of adulteration and so on. So that's the principle in a short for the random forest. And the other one, just have a little insight in there. So that works completely different, this method. So here, this method is inspired by the human brain. And the important thing is the neuron you see in these circles. So we start with our features. So that's the input layer, where, as I said, we have 37 features. Then we have these two hidden layers with 51 and 96 neurons. And then at the end, we have an output layer with servant classes. So that's the classes we want to predict. So pure cactus, pure sunflower, and all the adulteration in between. As the others said before, we split out our data. So we have 80% for training and 20% for testing. And then we compared this both method, how they perform in this specific case. And we use therefore the accuracy, so that's the number or the percentage, how many observations were correct classified. And yes, again, you see in the first column the number of observations, then the results of the random forest, and then of the neural network. And it's quite clear to see that at the random forest one, we have an increase. So the more data we sample from our distribution, the better the accuracy became. Starting from around 84% to 94%. And the neural network behaves a little bit different. So there we have more stable results. So it's already 92 at 1,000 samples, but then also just only reach 93% in total. And so we think at this point that both models would be suitable to use for this classification. There are some differences, but yeah, at least from 10,000 observations onwards, we have quite good classification results. And then the natural question we ask us is, yeah, how is it now in the real world situation? As I said before, we just use the simulated data because of the few data points. And now we asked our project partner to prepare also real world adulterations. So he prepared 33 of them. And then we used the models we trained with the simulated data and how much of the observed adulteration the models can predict correctly. And that's the results. So what is, not as we expected, but as we expected is that the accuracies are now lower than before. That's a typical behavior. So if you use real world data, then often the accuracy is lower. But what we also see is now a difference between the two models. So the random forest in this case is quite better than the neural network. And it reached even 90%, which is only 4% lower than before. So from our point of view, a very good result for the random forest, or an advantage of the random forest method. Let's look a little bit deeper in this. So here you see the confusion matrix, something what is very, very good to illustrate how the performance looks like. So we have left the random forest, and on the right one side, the neural network. And on the y-axis, you see the true labels, and on the x-axis, it's the predicted ones. So, and if you look now in a specific cell, so let's start here over there. So there's the cactus oil, and in total there were 27 cactus oils, so 23 plus 4. And of this 27 cactus oils, 23 were classified correctly. So all the numbers which are on the diagonal, that's the correct classified samples. And if you have numbers apart the diagonal, then that's the samples or the observations which were not classified correctly. So for example, with the 4 over there, so you have it is pure cactus oil, but it was classified as a mixture of 1% of adulteration. And so it's quite clear that here the random forest shows very good performance. It has some problems with adjacency classes. So for example, over there, so if the mixture is very close to each other, so between 7% of mixture or 9% of mixture, then it's the model has problem to detect it correctly. And the neural network is also good, but it has more problems with these adjacent categories. Yeah, to sum up, so 2% seem to be a little bit close. So if we would do it again, we would recommend that we use larger classes. So then it would be easier for the models to detect the differences between the mixtures. And the random forest performs here better. One big advantage of the random forest is that we can look in the at the future importance. So that only works because what I showed you before, we split on feature levels. So we can also make interpretation about that. And here it's the ranking of the features that split the nodes in a way that pure nodes are generated. So the idea is if you're in this tree structure, then a pure node is a node where only one kind of observation is obtained. So only cactus oil, for example, or only mixture 1% and 99%. And so in this case, we see that alpha tocopherol has the largest feature importance in that way that it generates this pure notes. So to sum up, we tried to show here possibilities how to deal with few data and classification model choice. And in this, let's say, project case study, it seemed to be that a combination of simulation and then evaluation on real-world data works very well to decide what model is a good choice. I would recommend in this specific case that the Monte Carlo simulation works very well compared to the CT gun, and also that the random forest with 1 million samples for classification works the best. And the advantage of the random forest is that we have this interpretability. So we can use this to interpret the output much better than in the other case with the neural network because of just how the method works. And so the final takeaway is that we expect that our model can detect with around 90% accuracy adulterated cactus oil from sunflower oil. And in this project, the traditional machine learning as random forest seem to have a good choice. So that would be kind of my last sentence. So there's a lot of discussion, I think, what method you should use. So a deep learning method or traditional, let's say, statistics or machine learning. And so here, at least in this project, it was clear that the random forest, which is a classical one, works very well or even a little bit better than the neural network. That's now my slide with involved person and acknowledgement. So this project was done at the AI consultancy unit of KIDA. So the team consists of me, Christina Ortiz-Cruz and Aaron Roggenland, and our partners at MRI, Versailles El Harkoui and Bernhard Matthäus. And the project was founded by the Federal Ministry of Food and Agriculture. So thank you very much, and happy to answer some questions.
Thank you. Thank you very much, Michael. Other questions? Katharina has one. I'm coming.
So maybe first, because it was in the beginning and it was quite fast, but maybe can you show again or tell again about the 37 factors you used? I think it was mainly fatty acids, right? Fatty acid pattern.
Yeah, it's fatty acid. I can show. Well, for that, I'm more in the field of the machine learning. So if you want to have detailed information about the 37 features, then I could make the contact with the colleagues. Yeah, but that's the measurement we used. And I think what, at least from our project partner, is different, that tocopherols are not so often analyzed in this field.
Okay, no, that's enough for me. It was just to the follow-up question. And maybe I'm not that much into chemistry, so maybe it's logic, but I'm gonna ask anyway. Wouldn't it just be possible to analyze the pure cactus oil and then get maybe ranges for those fatty acids and tocopherols, and then you could compare to any oil it would be adulterated with? I'm wondering why did you not just look at the cactus oil and define ranges, which, like, if you have a cactus oil, you usually have that fatty acid pattern, and then you can analyze any other oil or any adulterated oil, adulterated maybe with olive oil or whatever, and you could still maybe make an assumption about the authenticity. You understand my question?
I think so. Well, or I say something and you can say if it's answered your question. Well, so to be clear, I haven't shown it, but the cactus oil, it's not that it's one certain value, but you have also a kind of a distribution. So you have, for each feature, you have lower values and higher values. So if you put together with some adulteration with some other oils, then it has an influence. So if you have if you have a kind of range and then you put the other one again, and you don't know, well, if you buy this oil in a supermarket, for example, you don't know which which value has your specific oil. So you have these densities, and we wanted also a robust analysis or model, which if, for example, a company who does this adulteration knows that there's only one feature she has to change, let's say, and maybe somehow provide to look at it from the analysis, then it's quite easy. But if you have a more complex model where you have this the mean values and also the variance, so the dependency between the features, then it's much more difficult for a company to do this, because the results not only based on one specific feature, but on of the whole oil composition and also about the whole oil distribution. And so we choose this approach.
Thank you for the answer.
I was wondering, the only way that you can adulterate the oil is with sunflower oil. What instead of sunflower if they use the other kind of oil?
Yeah, that's let's say limitation. Yes, we use sunflower oil because it's often used, and also about this colorless. So there it's more difficult to see from outside. Well, it depends on the composition of the other oils. So if the composition is kind of, so the features, the attributes of the oil, if they are kind of similar to sunflower oil, for example, or at least far away from cactus oil, then our expectation would be that the model still works. Well, we have to try it out or need data for that. So that's a question of data. So if you have more data of other oils, then you could put it in the model and so on. Yeah, so that was, but the thinking was that it's the most used oil, and which is also you can't detect so easy. Yeah, and especially in this low ranges, so from 1% or 3%. And if you use other oils, then you might see it by eye, and then it's no problem anymore to detect it.
Okay, then it's obvious then, right?
Yeah.
Okay, do you have any questions anymore? Sabrina.
Thank you. I have two questions. One regarding the heat map you've shown about the predicted accuracy and the true positives for each class. If I understood it correctly, this was for the real life or real test data set where you get the.
Yes.
Did you look also at this map for the sampled test data set, and did you see there some kind of overfitting already for the neural network, or wasn't that obvious? Because or do you think that this might be due to overfitting of the neural network to the training data?
Yeah, so first, yes, we looked at the other confusion matrix as well. Well, the numbers are just a lot of higher, of course, because if you have 1 million samples, then you have high numbers, so it's not so nice to display. Yeah, the problem of overfitting, yes, we should look more in detail in this part. The thing is that in the when the decline in the accuracy arises, is it's at the point where we use the real data. And if in another one where we use the tested one, it was similar. So there we already had a test part included. So if there would be a lot of overfitting, we should see it already there, hopefully. And there it was not visible. So yes, we have to think about what is a good explanation why this changed to the other one, so to the real one. But yeah, one would be that it's that the other model, well, it just performs a little bit better. Well, overfitting maybe for the others is that the model learns too precisely the single data points, so that the neural network maybe not learns the structure of the system, but to learn the exact data points. And yes, that might happen. Yeah, that would be further research.
Because I was thinking if that's the reason, maybe it adopted more to the simulation process, that it captured the process how the data was simulated maybe better. if this would even be possible. I don't know.
Which part you consider to to the
neural
classification or
network,
simulation?
yeah, because the training and the test data were both from the simulated data, right? So there was a process behind it that got to the data, and it was the same for the test set and for the training set. But for the real data, there wasn't this process. So maybe the neural network captured this simulation process, if you know what I mean. Maybe it's a silly question, but
Yes, no, I think it's not a silly question. It's a good question, and I think maybe you found the answer already. So I think that could be also one part of the explanation. So that it's more concentrated on the simulation than on the and well that's a problem. Yeah, definitely. Yeah, thanks for this explanation.
thank you. If we have time, I would just one more technical question, just because I'm curious. For the random forest, did you do the hyperparameter training also for each of the number of samples you simulated? So did you do the hyperparameter tuning for 1,000 samples and then again for 10,000 samples, or did you just go with the out-of-the-box model? Or
Yeah, that's yeah. So you have the good questions. So yeah, we did hyperparameter tuning for 1,000 and 10,000, but then from 100,000 onwards, we had some computational difficulties because then it becomes very hard to do this hyperparameter tuning, and so we decided to stop with the tuning at 10,000 and then use this setting for the other ones. So yes, that's also one how it is, but yeah, we already had a quite hard or quite HPC, and it was not possible to do the hyper tuning for such much samples.
yeah, thank you. Yeah, that's the reason maybe I asked, because we ran into those problems often where we don't have the computational capacity. Yeah, thank you.
Yes, so but the thing from a let's say from a model perspective, when we look at the results, so there was kind of we stopped at 10,000, but so it was still that it doesn't become worse at least. So that is seemed to be that it's not the best, not the worst choice to do this, since because otherwise, if it would be a really hard influence, then it should decline very much the numbers afterwards. So it seemed to be that it's reasonable, but of course it would be better if you have more machine power available to also do the tuning for the higher classes. And I'm happy to maybe we can have later talk. So then.
Very good. Shall I leave you for coffee, or is there any question left in the room? Okay. The slides are going to be shared, Andrea? Yes. Okay. Okay. Wonderful. Okay. Well, then I'd like to tell you that sadly the last session is 3:30 in the big room over there, and I think in the meantime we'll have coffee. And thank you very much, our presenters, for today's session, and thank you very much for your questions. And have a nice afternoon then.